Respiration review video. This is brought to you by Maya and Gabe. So just as a quick roadmap of what we're going to cover in this video, there's basically 5 main sections which comprise of cell respiration and just the overview, glycolysis, the Krebs cycle, the electron transport chain, and how mitochondria play a role in all of this. So just as a quick overview, once again, cell respiration starts with glycolysis. Then if there is oxygen, then it goes through the link reaction, through the Krebs cycle, and into the electron transport chain. Meanwhile, if there's no oxygen, you move on to fermentation. This is a quick list of all of the understandings we'll cover in this video. I'm not going to dwell too long on it or read them all out, but these are all of the learning outcomes and understandings for section 8.2 for HL. So first of all, the introduction concepts. Cell respiration begins with glycolysis, and this is in the cytoplasm. Fermentation is also in the cytoplasm, and these are both of the anaerobic processes, where basically they don't need oxygen. Meanwhile, if you do have oxygen, you can create more ATP. So this is done through the link reaction, the Krebs cycle, and the electron transport chain. So first of all, some vocabulary that you need to understand the learning or comprehend cell respiration. One of these is repeating chemical reactions that keep occurring, because cell respiration is a metabolic process. This means it's catalyzed by enzymes and it's going through a whole bunch of chemical reactions. So 2 of the most important ones that pertain to hydrogen in particular are oxidation and reduction. And you might remember the mnemonic oil rig that Mr. Dubé lovingly taught us, which means oxidation is loss and reduction is gain. What are we losing exactly? In this case, it's electrons. These always occur together because they are opposites, right? So if one molecule is gaining an ion, the other one is losing it, and vice versa. Why? Because they're complementary processes. One is the loss and one is the gain of electrons. So basically, for the remainder of this video, you need to keep in mind in which step be aware of what is being oxidized and what is being reduced, because with one comes the other. So now we're going to talk about phosphorylation. And phosphorylation is another repeated chemical process, but this has to do with a phosphate molecule instead of hydrogen, and this is just the addition of a phosphate molecule. Now, what does this do exactly? It makes molecules less stable and more likely to react. One example of this, so you'll see this referred to as sometimes activation, sometimes phosphorylation, and in the case of ADP to ATP, you will see it's oxidative phosphorylation. This is called oxidative phosphorylation because the energy to synthesize this ATP or phosphorylate is derived from the oxidation of hydrogen carriers. So this is a quick summary of reactions that I won't really go through it, but it's a really useful thing to have in your notes. It basically separates all of the processes in cell respiration and what occurs in them, so be it decarboxylation, oxidation, or phosphorylation. So first comes glycolysis. And big picture, glycolysis is the conversion of glucose to pyruvate. More specifically, you're taking your glucose molecule and 2 ATP molecules, and you're converting it to 2 pyruvate molecules and an energy carrier called NADH. And 4 ATP are synthesized, leading to a net gain of 2 ATP. So the first step in glycolysis is phosphorylation. Glucose is phosphorylated by 2 ATP molecules into hexose bisphosphate. Glucose is becoming more reactive. Next comes lysis, which means the breaking apart. So the hexose bisphosphate is broken apart into 2 triose phosphates, aka 3 carbon sugars. Third comes oxidation. So these 2 molecules of 3 carbon sugars are oxidized. This means after this they become pyruvate. And along with this, as they are being oxidized, NAD plus is reduced into NADH. So that is the pair of reactions. While the 3 carbon sugars are becoming oxidized, NAD plus is picking up that ion and becoming NADH. Finally, ATP synthesis occurs, so the energy from all of those steps leads to 4 ATP molecules. Ultimately, some properties of glycolysis that often come up in either multiple choice questions or even sometimes they're important to mention in long answer is that it's anaerobic. You have a net gain of 2 ATP molecules, and your product at the end of this is pyruvate. So next, we're going to focus on the fate of pyruvate, as Mr. Dubé used to say. So when there's oxygen, it will continue into the aerobic processes, which include the link reaction, the Krebs cycle, and glycolysis. And without oxygen, it will move on to the anaerobic process, which is fermentation, which we won't get into in this particular chapter, but you can review it in, I believe, 2.8 or 2.9. So the link reaction, the only learning outcome related to this is that you need to know that pyruvate is being converted into acetyl coenzyme A through a series of reactions. So in order to remember link reaction big picture, just think it's linking glycolysis to the Krebs cycle. It's changing the location. So it's linking the cytoplasm, and you're going into the mitochondria now, where the next aerobic processes take place. And ultimately, the reactants are 2 molecules of pyruvate, and the resultant is 2 molecules of acetyl-CoA and 2 molecules of NADH. So first, your pyruvate is decarboxylated, which means it loses CO2. Secondly, your pyruvate loses 2 hydrogen atoms so this is eventually, this comes up later in the electron transport chain by creating another energy carrier, which is NADH. Thirdly, pyruvate, which is now called an acetyl compound, gains the coenzyme A, and this forms acetyl coenzyme A, and this is the reactant of the Krebs cycle. So this is how the link reaction links glycolysis in the Krebs cycle, because the Krebs cycle doesn't take in pyruvate directly. Rather, the link reaction needs to prime it to do so by turning it into acetyl-CoA. So the Krebs cycle, you have 2 molecules of acetyl-CoA, and this leads to 4 molecules of carbon dioxide, 2 molecules of ATP, 2 molecules of FADH2, which is another energy carrier, and 6 molecules of NADH, and 6 molecules of hydrogen, or 6 ions of hydrogen as well. So for just one acetyl coenzyme A, the first step is that an acetyl group is lost, and this goes back into the link reaction. It goes back out into the cytoplasm for the link reaction to occur again. Next, this reacts with the 4-carbon compound already present. So now your acetyl-CoA has transferred into a 6-carbon compound, which is often referred to as citrate. And 2 things happen to this compound. First, you lose carbon, and this leads to the formation of CO2, which is expired through your respiratory system. Then you have 2 hydrogens that are lost, and these hydrogen ions go to your energy carriers, which eventually make it to the electron transport chain. So now that has happened once, you now have a 5-carbon compound. And with this 5-carbon compound, those 2 reactions occur again, and also ATP is produced into usable energy as well. After all of these reactions, the 5-carbon compound is now a 4-carbon compound. This combines with the acetyl coenzyme A to create a 6-carbon compound once again. So this is why the Krebs cycle is a cycle, because you start with acetyl coenzyme A losing its acetyl group and being brought in with a 4-carbon compound to create a 6-carbon compound. Eventually this is worn down back into a 4-carbon compound, which can then be used again for any upcoming acetyl-CoA. So as an overview for the Krebs cycle, all of the processes that I just described are for one molecule of acetyl coenzyme A. So in fact, this happens twice per glucose molecule. So each glucose is broken down into 2 molecules of pyruvate, which is then converted into 2 molecules of acetyl-CoA. Then all of these processes happen twice. So you end up with 2 molecules of NADH, you end up with 4 molecules of CO2, and 2 molecules of FADH, and 2 molecules of ATP when you're moving from the 5-carbon compound to the 4-carbon compound. So now we're at the electron transport chain. As an overview, the electron transport chain occurs on the inner mitochondrial membrane, and the cristae increase the surface area for this to occur. This is one of the ways in which mitochondria's form is connected to its purpose. So as an overview, hydrogen carriers, so everything that we've created, be it NADH or FADH2, these are reduced by the electron transport chain, and this eventually leads to the synthesis of ATP. This process is called oxidative phosphorylation, as you're using oxidation reactions to get energy to phosphorylate ADP into ATP. So first the proton pumps create an electrochemical gradient. Then ATP synthase uses the diffusion of these protons to synthesize ATP. Ultimately, oxygen accepts the leftover electrons and protons in order to form water. So here's a quick diagram. You can take this time to copy it, or you can watch the video that I've created that shows this diagram in motion. Okay, so here you see an electron going through the first complex, and with this, it's losing energy. Therefore, it's allowing active transport to occur from a lower concentration of hydrogen to a higher concentration. And as it goes through each, it pumps up more and more, travels through the quinine, travels through complex 3, travels through cytosine C, travels through complex 4, and finally leaves. Meanwhile, all of these hydrogens are building up. All of these protons, rather, are building up. And therefore, they're allowed to diffuse through complex 5, or better known as ATP synthase. This allows energy. This catalyzes the reaction of ADP and PI to create ATP, the final end product.